I'm happy to be here, I guess. Yeah, it doesn't take too much time to make the journey. So I guess today will be a little bit different than what you're used to, or at least maybe you're expecting it to be a little bit different, but a lot of the same sort of principles that maybe you've come accustomed to hearing and thinking about, I hope to convince you is actually pretty widespread across the kingdom of life. And so I'm going to start just to tell you a little bit about sort of the outline of how I have this talk organized. I'll give you a little bit of background on some things you may need to know so we can all kind of be on the same page. And then I'll tell you a little bit about, just a little bit about some past research already published from my postdoc. And then I'll jump into sort of what I'm trying to finish up now. Also work that I started from my postdoc I need to mention. And then hopefully, if we have time, I'll be able to tell you a little bit about some of the stuff that the lab has started doing over the last 8 months. All right, so I think like a lot of people in this room and in your department are really concerned with trying to understand the relationship between genotype and phenotype. So plants, like other eukaryotes and multicellular organisms, have a lot of intraspecific variation, right? So here's some examples of Arabidopsis in this bottom in the middle of this slide here. This is all the same species. You have different morphologies. We have different colors, different architectures of the plants, different total shapes. And so there's a lot of intraspecific phenotypic variation that exists within species also in plants, right? And also like humans, when we use GWAS to try to understand that association, we find that most of the trait-associated genetic variants are located in non-coding regions. So up to about 95%, depending on what species we're talking about, of those trait-associated variants end up being in non-coding regions of the genome. And so if we want, yeah, so far so good, right? So far so good. If we want to understand how those variants are impacting phenotypes, what is the link, right? We need to understand a little bit about potential mechanisms. And one of those ways, I think, lucky for me, I've always been interested in cis-regulatory elements, and this is sort of a justification on studying them further. But cis-regulatory elements, let's just get, if you don't think about those as often as me, I just want to give you a quick background. And so generally, they're known as binding sites for sequence-specific transcription factors that have some sort of general impact on transcription. And so there's a bunch of different flavors of cis-regulatory elements. Probably the best understood are promoter elements. So those are the elements that are located just upstream of transcription start sites. I'm trying to point to it here. So that would be a promoter sequence. Those have been leveraged a whole bunch in plants to make reporter lines and promoter bashing to look at changes in cell type expression patterns. So a lot has been studied on those, and they're easy to identify because of their location relative to genes. Somewhat less well understood are enhancers, which can be hundreds of kilobases away from the genes they regulate. They can be upstream or downstream or even within the genes themselves, particularly within introns. And they operate in sort of a location, orientation, and position independent manner of the genes they regulate. And then sort of the last two are silencers, which, as their name implies, elements that silence gene expression. And we also have insulator elements. And these are elements that basically demarcate and restrict the area in which different regulatory elements can act. And so all of these different classifications of cis-regulatory elements also exist in plants. We have evidence for insulators and silencers, promoters, enhancers. And they can have huge impacts on phenotypes. And probably my favorite example is this one of maize. So maize is corn. If you go to the grocery store and you buy corn, this is a species that you take home. So corn comes from a subspecies of maize known as teosinte that originated in central Mexico. And teosinte is this plant on the left side here. And there's several differences, but one of the major ones I hope is readily obvious is that this one, teosinte, makes tons and tons of branches, right? And modern maize is only a single stalk with apical dominance. And so one of the very fundamental changes that led to the modern maze that we see now from teosinte was this insertion of a transposon upstream of a gene called TB1. And so this transposon confers enhancer activity to TB1. So in teosinte, the insertion is absent in this region, and you get very low levels of expression of TB1 and almost no expression in the axillary meristems, which are these stem cells that exist in the axils of these leaves. And so when you have high levels of expression in the axillary meristems, TB1 suppresses lateral branch formation. So this is just one example. And I should also mention that this insertion upstream of TB1 is fixed in all of modern maze. And if we delete this regulatory element that's located on hopscotch, you can revert back to the ancestral phenotype, right? So this is one example of a single transposon, a single regulatory element variant that has a pretty drastic effect on the phenotype. All right, so how do we identify regulatory elements genome-wide? There's a bunch of different ways. Over the years, I've counted things like DNA sequencing, MNA sequencing. FAIR-seq had its time in the limelight for a little bit, but I would argue that probably the most dominant form of cis-regulatory element profiling nowadays comes with ATAC-seq. And so ATAC-seq stands for Assay for Transposase Accessible Chromatin Sequencing. And all of those methods, I should point out, sort of leverage the biochemical feature of regulatory elements in that typically they require accessible chromatin for activity, right? So a transcription factor, most transcription factors can only bind regulatory elements that are located within accessible chromatin. And so the way that ATAC-seq works is we have this hyperactive transposase, which has sequencing adapters loaded to it, forward and reverse sequencing adapters. And so when we treat chromatin with this transposase, we get preferential integration of those adapters within accessible chromatin, right, shown here. And conveniently, those adapters are known. And so we can amplify fragments with forward and reverse primers to recognize those adapters. And so when we get a final library product that looks something like this, right? So then if we sequence that library and align those reads to the genome, more specifically, if we look at the ends of those fragments or the starts of the reads and do a pileup of those genomic coordinates, we find things that look like this, which we're going to call accessible chromatin regions, right? So these are regions of the genome that show high levels of accessibility and putative regulatory activity. All right, so when I started my postdoc, there was this motivation to try to understand genotype and phenotype relationship. But sort of the sub aims of those was trying to understand more of what are like how does molecular diversity result in cellular diversity? And so one of the things that we didn't know at the time was how regulatory elements were involved in the specification, maintenance, and also differentiation of plant cell identities. And then sort of generally, we also don't know, we have a very limited understanding of how regulatory element variants affect cellular and molecular and also organismal traits outside of those few case examples that I've shown you. And the issue was that we don't have stable plant cell lines, right? So unlike mammals, we can't just culture differentiated plant cells indefinitely. They will de-differentiate, they become totipotent and become just a mess of stem cells, which is a cool biological phenomenon, but it makes studying cell type specific processes and functions really impractical. And so that's a huge challenge. And so when there have been efforts to profile and understand cell type specificity and cell type functions in plants, it's been really hard. And the way that it's had to have been done has only been done actually in a single tissue in one species, in Arabidopsis roots. And that was a huge effort where basically they had to use known marker genes, right, with known cell type expression patterns and clone them in front of a reporter and then sort cells, right? So you're taking the promoters of some well-characterized marker gene, attaching it to something like GFP and then sorting cells, and then doing molecular profiling on those purified cells. The other way was through just manual isolation, like manually taking one cell at a time or using something like laser capture, right? Yeah. Yeah, it was rough. And so sort of the synthesis of those issues is, all the regulatory element research that has been done in plants prior to this work has been limited to bulk or tissue scale sort of profiling, right? And that's not ideal because plant tissues are homogeneous populations of cells, sometimes up to 30 different types of cells in a very limited small space. All right, so that's not great. All right, so now I'm going to tell you about some of the ways we're trying to address those challenges. And like I hope most of you are aware of single cell profiling. This is becoming somewhat obsolete now, but I still feel like I have to. Usually, I give this talk to plant scientists, and they still haven't heard of it, and so I have to include this slide. Maybe this is something everyone else is really familiar with, but in case you're not. The way it works is we'll isolate nuclei, right? So we'll digest, release nuclei from cells either by chopping the cells with a razor blade or grinding, pestle and mortar. So then we'll isolate nuclei, and then we'll tagment the nuclei with that transposase that's loaded with sequencing adapters. And then the magic happens where we load the nuclei onto this microfluidics chip, and we get a stream of beads and the stream of nuclei that move through the channel. And then there's a stream of oil that will partition droplets. And so the idea is that if we can get a single nucleus and a single barcoded bead into one droplet, then we can profile the accessibility of that nucleus. And so each of these beads contains a 16-mer oligo that's appended to all the accessible regions from the nucleus that's in that droplet, right? So if you can just keep your focus on this one droplet here, it has one colored bead and one nucleus, all the accessible regions that have already been tagmented in the genome will get a short 16-base barcode attached to them so that we can tell later down the line after we do sequencing that this read came from some nucleus, right? We can put all the reads together that have the same barcode, and so we know that all those reads come from the same nucleus. All right, so we applied that method to 6 different organs at different developmental stages in maize. At the time, there was a lot of nuclei. Now people will probably laugh at that because we have some atlases coming out with the tens of millions of cells across various stages of development and the life cycle of organisms. But at the time, this was a pretty big effort. And so one of the first things that we noticed, first of all, is that cell identity, so each dot in these plots represents a single nucleus. And sort of generally, dots that are close together have more similar chromatin accessibility profiles across the genome. So 2 dots that are close together are more similar. 2 dots that are very far apart, generally not as similar, right? And so one of the first things you'll notice is that cells that come from the same tissue look more similar to each other, right? So there's a spatial sort of spatial information that's captured by doing single cell profiling. A lot of the cells that come from the same tissue share a lot of their accessibility profiles. Although we do see some intermixing of cells from different tissues. All right, so generally we find about 10 large, what we're going to call clusters. But when we zoom in on those major clusters, we find additional sort of heterogeneity amongst different cells. And so it was really that level of resolution that is what's denoting the cell types in our data sets. So just to give you an example of that, I'm showing you on the left side 2 genes that are specific to bundle sheath. It's a vascular cell type, makes up the veins in the stems and in the leaves. And they're part of the photosynthetic machinery. They partition C4 photosynthesis. We don't have to get into that. But the point is that these 2 genes here are specific to this cell type. And so if we look at the accessibility of these 2 genes within across different cells, we can see that we have co-localization of signal in the same population of cells. Right? So the same cells are showing specificity of chromatin, increased levels of accessibility for those 2 genes. Likewise, we have 2 genes that are specific to mesophyll cells, and if we look which cells show high levels of accessibility for those 2 genes, we see again co-localization and overlap, right? So we know that these 2 genes are specific to mesophyll, and we see the same cells show high levels of accessibility. And I'll just point out that we're using accessibility here as a proxy for gene expression. Yeah, Yeah, I think it's fine.
Sorry. So I was going to say, I'm not a plant person, but maize has a lot of duplications in it, right? Like a lot of plants. So how does that, when you're actually functionally mapping the ATAC reads back, how do you distinguish between the different copies and so forth? Or do you just throw those out and this is all based off just the unique mappings? Or I don't understand. It feels like that would be a big challenge.
Yeah.
Clearly you've overcome it. I'm just curious how.
Yeah, that's a great question. So the way I handle that, so maize does have a whole genome duplication event. Thankfully, it was long enough ago that one of the genomes has become like a subgenome, and there's subgenome dominance in maize. And so one of the two copies got destroyed basically, but it is still an issue. And so the way we handle that is we set high mapping quality thresholds, and then also we remove reads that have multiple mapping or even potential other mapping, if that makes sense. So it does. It's the XA flag. I don't know how else to say it from BWA, but if it has an XA flag, we get rid of it.
So heavy filtering, but you still have enough signal to be able to get.
Yeah. As long as we're using unique reads, I think that's, to me, I feel pretty confident. But yeah, so we'll use chromatin accessibility sort of a proxy for gene expression, and I mean we can show it this way on the UMaps, right, by using a sort of heat as signal. But we can also look at it in a more conventional way, which would be to take all the cells from an individual cluster and pile them up, right? And so in this way, you can see that only one of these clusters, cluster 38, shows any sort of specificity and accessibility upstream of these two genes, which end up having to be specific to xylem. And so we can, using this sort of approach plus some automated machine learning based annotation approaches, we got about 52 different types of cells in this data set. All right, so it's great to be able to identify cell types, so we have to use it for something useful. And so one of the things that we wanted to know right away is, first of all, how good are our annotations? And then number two, can we make predictions on gene expression for genes that we actually have no idea what they do, what their expression profiles look like? And so we teamed up with Dr. Andrea Galavoti's lab at Rutgers, along with Zhonglian Chen, who was very kind to do some RNA in situ hybridization for us. So we gave them a bunch of genes, just showing you one example here. It's the GRF transcription factor that we predicted would be highly expressed or would be specifically expressed within the inflorescence meristem of the maize tassel, which is this part up here, as well as in the spikelet meristems, which are these cells down here, and also zoomed up on this side. And lo and behold, our predictions matched exactly what we saw with the RNA in situ. And so even the magnitude, we would predict higher levels of expression within the inflorescence meristem, which is this part up here, which we see happen. All right, so we started to feel a lot better about our data set and actually its use for being able to generate more markers in the future. All right, but because we're focused on regulatory activity, we can't just look at accessibility, right? We also have to look at gene expression and transcriptome profiling. And so we also generated single nucleus RNA-seq data from maize seedlings, so it's a very young stage of the seedling, and then did an integration with Liger. I don't see Josh here. I was going to make a joke about Liger, but he's not here. Yeah, bummer. But we used Josh's software to do an integration, and it worked really well. We can capture all the different cell types, and we get very good overlap and consistency between these two different modalities. Just to show you kind of how well it works, I'm showing you 5 different marker genes, as well as the cells that are specific to the attack modality and cells that are specific to the RNA profiling. And we see very strong overlap between signal for those two different profiling methods. All right, so how can we use these data to learn something new, right? I mean, it's great to, just getting cell type specificity to all in plants is kind of a big deal because it's just something we don't have. But can we learn something new about the biology using this? And so one of the things that I noticed is that when we compare sort of generally gene accessibility with expression across these different cell contexts, we get a very good level of agreement. It's about 0.6 correlation for Spearman's correlation. But one of the things that you notice is that in some instances, we see genes that are highly accessible, but they lack expression altogether. And that to me was kind of weird, right? It's like, how do we explain this discordance between the correlations, right? We have good correlation, but it's not one, right? So obviously there's something else happening. And so when we take these genes that are accessible yet silenced, one of the things that we notice is that they're highly enriched for K27 trimethylation. And so in plants, and I think this is also the case in mammalian systems, but K27 is a classic mark of developmental gene silencing. And what was really interesting is that most people actually thought that K27 was associated, trimethylation is associated with like closed chromatin, inaccessible, you know, I mean, obviously you're not being transcribed, but the idea is that these are just knots of chromatin all wound up. And that's actually just not the case. We see high levels of accessibility in promoters, obviously not as high as expressed genes, but notably higher levels of accessibility compared to genes that are marked by DNA methylation, for example. And we also noticed, we're wondering how can this be? And so we were looking at the accessible regions that are specific to the silenced genes that still sort of maintain accessibility, as well as have K27 trimethylation, and we found these polycomb response elements within their promoters. All right, so these are TF binding sites for transcription factors that recruit the PRC2 repressive complex. And so this is actually one of the first instances, at least in the plant, that shows that you have to have some level of accessibility in order to facilitate silencing. And that actually still somewhat maintains an accessible configuration. And that allows the PRC2 complex to come in and silence those genes. So it's not just a ball of wound up chromatin. There's actually some fluidity and accessibility there. All right, so another way, we realized, and this is unpublished, that we could use these data was to try to do a sort of in silico ChIP-seq. And this is kind of a big deal in plants because we don't have antibodies for transcription factors. I think there's like 50 antibodies total for transcription factors in all plants. And so if you want to know where your transcription factor of interest that you've been studying for decades is binding, well, good luck, right? But we realized that we could use these data to sort of make predictions about that. And so the way that it works is we'll start with a fully integrated, and actually this would be greatly improved by using the multi-omic profiling methods that exist now. But what we did was we took this integrated embedding and then did a reciprocal imputation. So we imputed the RNA profiles onto the attack cells and then imputed the attack accessibility data onto the transcriptome cells. And so what that allows you to do is to do an association test between the level of expression of a given transcription factor and the accessibility at any given locus in the genome, right? And so you could do this in a specific, like in very specific cellular context. So you can split up your cell types and run the association. You see the association disappear in different cellular contexts. Maybe the genes never express at all, in which case you'll not find an association, right? And so when you do that, you can identify all the regions of the genome that have a strong positive association between the level of accessibility and the expression of some transcription factor. And when we take those regions, we're actually able to find and capture the motifs that you would have predicted. So for example, the transcription factor that we profiled here is a BZIP61, and the motif that we recovered de novo looks just like another BZIP69 in Arabidopsis that has been profiled using DAP-seq. And so this is a way that we can very quickly profile and make predictions on interactions of transcription factors with different regions of the genome without ever having to do an experiment. And you can, of course, you could do this for all transcription factors in a genome. All right, so the last thing I want to talk about as far as published data, to me was actually one of the more exciting results. And so one of the things that we did was we're just curious, in trying to establish the functionality of these regulatory regions. One of the things you can do is look at the level of selection or the level of DNA polymorphisms that exists in those regions. And so we compared the level of SNP enrichment, all the variants that exist intraspecifically within the species, within cell type specific ACRs, accessible chromatin regions, and also like the non-cell type specific accessible chromatin regions. And actually what we find is very high levels of conservation or depletion of variation within cell type specific accessible regions. But when there is genetic variance within those loci, they tend to have big impacts on phenotypes, right? So here I'm showing you the level of enrichment of GWAS hits in both of those scenarios, right? So despite being under lots of strong purifying selection, cell type specific accessible regions, when you do have variance in them, they tend to lead to phenotypes that can be captured. All right, so now I'm going to tell you and hopefully convince you a little bit, convince you to join me in studying plants. And this was actually a strong motivator for me in realizing how feasible this project I'm about to tell you about really was. So we use maize for trying to understand molecular variation, and here's why it's amazing. Number one, it has very rapid LD decay. In humans or mouse, you would need 10 to 40 times as many individuals to get the same resolution, the same haplotype size, to be able to find map variants that are causal. Okay, so we need a massively reduced population size, and that makes the experiments much more feasible, at least from a cost perspective. There's also extensive just nucleotide diversity. We have lots of variation within the species, and you need variation in order to map these traits, right? We have lots of phenotypic diversity. These are all the same species, right? They can, the female inflorescence look totally different, and the plant architecture of the stalk and how many leaves and what they look like is very diverse. And we actually were the first species to have multiple whole genome reference scale assemblies, end-to-end telomere assemblies. And that was published back in 2021. So we had 27 genomes when the human genome still only had one. So we have lots of resources available to us. All right, now that we're all going to be maize geneticists moving forward, I'll tell you about the experiment. So what we did, and this is with the help of Xuan Zhang in my postdoc advisor lab, Bob Schmitz. And what we did was we had this population of 172 very distinct maize genotypes geographically spread throughout the entire world. And we took nuclei from anywhere from 4 to 27 genotypes at once, right? So we'll take seedling tissue, put it on a petri dish from all these different genotypes, chop them up, release nuclei, do single cell attack-seq. And because all these genotypes have already been sequenced, we can use the known genotypes, SNP data, VCFs, to go back and say, okay, does this cell look more like this genotype A? Does it look more like genotype B? So we use maximum likelihood to assign genotypes just sort of ad hoc, right? And so this is a way we can very quickly generate lots of data without having to do nuclei isolation one genotype at a time. And the other benefit to this was that we realized we could massively overload the chip and generate lots of data, kind of skirting the recommendations that are recommended by the manufacturer, because we knew that we could remove the barcodes that represented doublets in our data set. We can talk more about that later, but just to give you a sort of overview of what it looks like, one of the things that I was curious to understand is just what kind of variation exists within the species in terms of regulatory regions. So we can identify these highly conserved accessible regions where the sequences themselves are highly conserved. I told you that we have whole genome sequencing for all these inbred lines. We can find also regions where the sequences themselves are dispensable. They're lost and not totally present across all the different inbred lines in this population. So sort of altogether, we found about 70% of accessible regions, the sequences themselves are conserved within the species. And that's also supported by conservation across different plant species within the grasses. And so we feel pretty confident these are very highly conserved, and other regions that do show regulatory activity tend to be more variable within this population. All right, so just digging into those a little bit more, we found that the conserved, this is not really that unexpected, but the conserved regulatory regions had a much lower overlap with transposable elements. So it seems like the dispensable or the newly evolved regulatory regions tend to arise, or at least are more associated with just transposons generally. And another sort of indication that things that are conserved are important is we can also map star-seq data back onto these regions, and we find that regions that tend to be more conserved have much higher levels of enhancer activity. And then sort of the nail of the coffin for me is that we can also train a model to see what are important predictors of conservation within an accessible region. And we find that a lot of transcription factor binding sites that are very core developmental transcription factor binding sites, like lateral boundary domains, squamosa, promoter binding proteins. So transcription factors that are important for development of the plant tend to be very highly conserved and contribute to conservation of an accessible region. Whereas transcription factors that are more responsive to the environment, such as DREBs, so these are drought response elements. Worky transcription factors are known pathogen response transcription factors, and so those tend to be less conserved, and these might be just more important for sort of immediate or local adaptation, and maybe not necessarily conservation, across the entire species. All right, so I think the obvious thing is that when you get lots of, you generate this molecular data and from a whole bunch of different genotypes, one of the things you can do is try to map those molecular traits. So here I'm showing you a single locus with a bunch of different genotypes, and we can find regions where we have, accessibility is highly dynamic. And it turns out that we can actually map this as a phenotype using a GWAS-like implementation. So we can generate Manhattan plots and identify variants that are associated with the variation in chromatin accessibility, right? So to me, this was really exciting because now we can start to get at what are the determinants of accessibility in plant species, which was unknown. And one of the first things that we wanted to check is just, do we capture known cis-regulatory variants that exist in the species? And indeed we do. So here's a few examples. CCT9 is a known regulator of flowering time in maize, and there's a very well characterized transposon insertion that conferred adaptation to longer day lengths. So maize came from the tropical regions, and then we brought it up into more temperate areas where we have much longer day lengths. And so that allowed maize to better withstand our climate. And so we see that specifically within temperate lines. We see accessibility, and you would infer also greater regulatory activity in lines that are in temperate areas. We also find some others, also flowering time related genes, and some others that are associated with differences in architecture. This is actually a fine mapped cis variant within a promoter that affects the angle of leaves relative to the stalk in maize. And so having very narrow leaves actually allows you to plant much denser, and it's a huge agronomic interest. So we were able to find some of these variants that have been previously characterized. One of the cooler things that I was actually pretty amazed to see was that when we take these chromatin accessibility QTL, we can compare them with known expression QTL. And we get about 25-ish% of accessibility QTL are also associated with expression variation. And it turns out that most of the alternate alleles in the population are deleterious, at least if you're a gene that wants to be expressed or a region of the genome that wants to be accessible. So most alternate alleles reduce gene expression, and most of them also similarly reduce accessibility. It's about 75% of all chromatin QTL. It also turns out that most of the large effects that we see come from rare alleles. And that's actually also consistent with eQTL data that exists in maize. So, and it seems like these rare alleles that are mostly biased, well, I'll tell you about the cell type profiling here in a second. But the takeaway here from this slide is that the rare variants tend to have larger effects on chromatin accessibility and gene expression. All right, and because we did single cell sequencing, we can also look at the cell context variability that exists in these data. So ultimately, we came out with about 700,000 nuclei. This was about a year and a half worth of data collection. And thinking I was done halfway through, and then trying to do cell type specific chromatin mapping and realizing I had a lot more to go. But thankfully, we finally finished data collection on this. And we can very nicely find all the different cell types based off of different marker genes, just like I described to you before. But now we have a much more comprehensive atlas, at least in terms of numbers of nuclei and ability to detect regulatory regions within maze. And just like the bulk scenario, we can also identify chromatin QTL in specific cell types. And this was actually quite interesting to us because it turns out that most of the chromatin QTL that we identify are very cell type specific. So for example, here's a QTL that we find in bulk tissue. And then when we look in different cell contexts, we only find this QTL present within mesophyll. So it turns out that the cell context is an important determinant of how these variants that affect accessibility are able to manifest. All right, so I told you that most chromatin QTL are very limited in the number of cell types that they're detected in. And the rationale for that, or at least our observation, is that it turns out that the patterns or at least the significance of chromatin QTL identification and the power to detect it very closely mirrors the actual patterns of accessibility, right? So we're able to define chromatin QTL that's specific to a cell context, and it also turns out that accessibility is very specific to that context as well. So most of the time we're finding chromatin QTL with cell type specificity because that accessible region is cell type specific. Whereas things that tend to be more broadly accessible, they tend to get sort of masked in the profiling. All right, and because we had lots and lots and lots of TN5 integrations, one of the things that you can do is look for these things called transcription factor footprint. And so transcription factor footprints are these basically regions where you have a transcription factor that's bound. And TN5, which is that hyperactive transposase, can't actually integrate its adapters where that transcription factor is bound to, right? So it can only insert adapters flanking a transcription factor. And so we used fine-mapped chromatin QTL to see if, well, do we find any variants that have very high effect within transcription factor footprints, right? So first of all, we used this transcription factor footprinting method to identify regions or small segments of DNA within accessible regions that we predict would be bound by transcription factors. And we find that more than 50% of these TF footprints or transcription factor footprints have fine-mapped chromatin QTL within them, right? So these are statistically fine-mapped using sort of the summary statistics downstream and taking into account LD. But more than 50% of them are located within TF footprints. Right? So we think that this is sort of the main causative, the mechanism for how chromatin accessibility is variable within the population. One of the things that we have to check, right, is that if of course, we're doing transcription factor footprinting in different cell contexts, that the footprints that you recover are reflective of that cell type's biology. And indeed, we find, for example, the lateral organ boundaries, transcription factor is enriched in the lateral organ primordial boundaries cells. We find others like the DOF transcription factors, which are known in specification of phloem cell types. We see those in the phloem, as well as in the phloem precursors. I'll save you sort of the plant biology of this slide and just tell you this is kind of what it looks like. But one of the things that we noticed, so here we're mapping this top box is the transcription factor footprinting score, which is output by the software that we used for this analysis. And sort of the way we did it to add power was to aggregate genotypes that share a haplotype in a very small region in order to really boost up the number of observations that we get. And so one of the things that we noticed is that the transcription factor footprints that tended to be cell type specific were on the edges of accessible regions. And sort of more generally, when we took that anecdotal observation and sort of mapped it out throughout the entire genome, we find that the transcription factor footprints with very high levels or that are cell type specific, we only find those footprints within one cell type, that they're located primarily on the edges of accessible regions. Whereas transcription factor footprints that we find in multiple cell types tend to be located in the centers or more evenly distributed throughout accessible regions. And so we hypothesized that maybe, it's the differences in accessibility in the flanks of an accessible region sort of generally that is what's conferring cell type specificity, right? So you have, an accessible region, and then you have a nucleosome gets kicked out. It certainly makes the accessible region a little bit bigger. Now you have a transcription factor binding site that's exposed to a transcription factor in the nucleus. And indeed, about 85% of accessible regions, the variance, so here dark color means more variability across cell context. Dark color, 85% of accessible regions, that variance is on the edges, right? A very small amount, about 15% is more located within the center. So most of the time, the variation in accessibility is happening sort of on the edges of an accessible region. And so we were wondering, okay, well, do we see any transcription factors that sort of show bias in where they're located? Do we see things that are almost always in the center or almost always on the peripheries? And we find this transcription factor TCP, this entire family, is highly preferentially located within the centers of ACRs. And I've also highlighted here a transcription factor called leafy, which is the only known pioneer factor in plants. So to me, this was kind of a curious observation, which stemmed the next test. So we took all those fine mapped QTL and we said, are there any transcription factor footprints when they have a QTL in them that have an outsized effect on overall accessibility of that region? And if we do an enrichment test, we find that, again, the TCP transcription factors, the entire family is highly enriched for having large effects on accessibility. And so it kind of gives us this model here where if you have a variant in a non-TCP transcription factor footprint, you might get a little bit of change in accessibility, but not much. Whereas if you have a variant in a TCP transcription factor binding site, it has a drastic effect on accessibility. And to us, this was really exciting because TCP, we don't know any pioneer factors outside of leafy in any plant species. And so trying to come up with, and this is something that my lab is interested in sort of following up on, is trying to map what are pioneer factors in plants. All right. I'm going to try to go over this really quickly, but we're also trying to find ways to democratize single cell approaches. One way you can do that is using combinatorial indexing. So this was work done with Xiaoyu Tu and Celine Zhang at the Chinese University of Hong Kong. Basically, it's combinatorial indexing, but we did it in plants. And it gives really good quality data, better than 10x. We have, and part of the trick here was that we used a modified TN5 enzyme. It's a little bit bulkier, much less likely to get spurious integrations. And so the gist takeaway from this is you get slightly higher signal to noise, which we measure by the proportion of reads that are within peaks or within accessible chromatin regions. We get about the same number of cells recovered. It's a lot cheaper. You don't need any proprietary methods or instruments to generate this data. But that kind of led the way for the next sort of innovation that Bob's lab and sort of what I hope to get help with, because I don't have as good of hands as Xuan. I used to make all the single cell libraries in lab, and then Xuan joined, and then once we saw the quality of his, I wasn't allowed to touch the chromium anymore. But so what Xuan and I came up with was a way to massively try, stemming from the observation that you could pull lots of genotypes together and deconvolute those genotypes later. We realized that we could just use the tagmentation reaction itself to uniquely index cells. So here and I should also mention here that this method, we weren't the first to do it. Darren Krusanovich's group in Arizona, their preprint came out about a month before us. And there are some subtle differences in how the method works, but it's principally the same. But the way it works is we'll uniquely label about 10,000 nuclei within an individual well using different combinations of forward and reverse barcodes on TN5, and then massively overload the 10x chip. So in this case, I'm showing you we did about 20,000 nuclei. We know now that we can go up to almost 400,000 nuclei. And so we're getting lots and lots of nuclei within droplets, but the chance that two nuclei share the same TN5 barcodes is very low, right? And so in this way, we can sort of double dip and get labeling much later, or get labeling as a second round that allows us to have much higher throughput. And so we apply this to different genotypes. So the rows here represent different genotypes of maize. I want to show you that we're able to first deconvolute those cells based off of their TN5 barcodes, right? So each well ID is known because of the combination of forward and reverse adapters. Then we can also look at, how well do those nuclei match our expectation on genetic variation, right? And so we get about 90% of nuclei that are perfect match to our exact expectation. And part of the reason it doesn't match perfectly is in addition to doublets or collisions, I should say, we also get a little bit of contamination from index hopping, which is kind of a headache with the newer flow cells. But data looks great. We can get about 100,000 cells from one 10x reaction, which is about a 10-fold. Now we know we can get up to a 20-fold increase on the throughput. We get slightly lower number of integrations per cell, but we get a higher signal to noise ratio. And this is also known for sequential rounds of barcoding, that those things tend to result in somewhat less or a loss of complexity. All right, so you learned some single cell stuff that makes, we're learning how to uncover more plant biology. I know we're all really fascinated by plants now, and we'll leave the room all wanting to study maize. I showed you that a lot of the trait variation the trait-associated genetic variation are located within cell type-specific accessible regions. A lot of sequence variation, particularly within regulatory regions in maize. The chromatin QTL are highly cell type specific. We think that these TCP transcription factors are acting as pioneer factors, but this is something my lab is following up on. And then of course there's this movement to try to democratize and make single cell experiments more affordable, because right now it's just not. And my startup knows it because it's been disappearing really quickly. But yeah, so I think I've taken up all my time. I definitely need to thank a bunch of people for supporting me and giving me a place and space to do science, and as well as actually helping with science. So Bob Schmitz was my advisor at University of Georgia. He just finished his HHMI interview, and he's, I hope, breathing normally again with a reduced heart rate. Schwan helped me a whole lot with library preps and has done a little bit of analysis. Our collaborators at Rutgers, who did all the in situ work, and then Celine and Xiao Yu at CUHK, they also helped with a lot of the technological innovation. And of course, my funding part of this was partially funded by NSF when I was a postdoc, and then also my K99R00s still funding some of this work. And then, of course, I have to thank Michigan for allowing me to have a lab and sort of gambling on me. So I guess with that, I'll take more questions if we have any.